Good afternoon, and welcome to today's lunch symposium sponsored by Concept Medical. I'm delighted to be here co-moderating with Dr. Scheinert, and we have an all-star panel, and we have a couple of late arrivals, I'm sure, will be joining us. To my right, we have Drs. Ramon Varco and Brian DeRobertis, and across the way there is Dr. Eddie Choke, and we should be joined by a couple of other panel members shortly. Today's program is going to dive deep into sirolimus-coated balloons, and we're going to start first by having Dr. Alok Finn explain to us what the first principles are, going back to the beginning, the importance of preclinical evidence and what the data indicates for MagicTouch Sirolimus-coated balloons.
Sahil, thank you. What a great introduction. Couldn't be happier to be here. Thank you, Dirk, for inviting me. Here are my disclosures. This is the current U.S. perspective FDA-approved DCB landscape. As you can see, all the DCBs that are approved for any indication in the United States all have one thing in common. They're all paclitaxel, and most of them are approved for above-the-knee indications. As you can see here, the first four are, and then you can see there's Agent, the only DCB in the United States that's been approved for coronary, and that's for coronary ISR. But sirolimus-coated balloons, I think, are gaining a lot of momentum, and you can see here that there are multiple studies that are ongoing or about to be presented, including the recently released Solution ISR trial, the Magical ISR trial involving the MagicTouch balloon, and then we've got some other de novo trials as well. So I think the landscape of sirolimus-coated balloons may overtake eventually, especially in some situations, the paclitaxel-coated balloons, and perhaps below the knee as well. You can see that paclitaxel formulation types, really the important thing to know is that all of the balloons I showed you in the previous slides share the makeup of crystalline paclitaxel. Crystals of paclitaxel basically are transferred to the vessel wall, and then that's dissolved over time to become amorphous and to lead to biological effectiveness. There's no doubt about biological effectiveness in terms of those balloons. They all have good PK results and good clinical results for the most part. Now, it must be said that one of the things that I think has received some attention, but not a lot of attention, and I think Sahil is talking about this tomorrow, is the fact that every paclitaxel balloon we've ever looked at, and these are all the commercially approved paclitaxel balloons in the United States I just highlighted for you, is associated with distal embolization. This is in a peripheral model in the porcine where we've deployed the balloons in the SFA of the animal, and then we look downstream at the skeletal muscle, and you can see what's interesting is that every balloon is associated with sections where we can find paclitaxel in downstream arterioles in the skeletal muscle, and you can see it really ranges in its percentages of sections from highest for impact, the most loaded dose of paclitaxel, to Ranger to Stellarex, and you can see earlier studies with Lutonix and impact are shown there. And just to back that claim up, you can see we also assay the downstream skeletal muscle for drug levels, and you can see that in some cases the drug levels are exceedingly high, up to 911 nanograms per milligram shown in the coronary band after impact, and you can see generally much higher in the coronary band, which is a vascular structure, than in the skeletal muscle. Now, why does this occur? How could this occur? This occurs because the range of sizes of particulates released by paclitaxel-coated balloons are large enough that they oftentimes lodge in small arterioles downstream. Doesn't matter whether you're working in the peripheral vascular beds or you're working in the coronary beds. You can see these no matter what type of downstream vascular bed you're looking at. And you can see these sirolimus-coated balloons in this particular case have an advantage because they are not composed of crystalline sirolimus. They're actually composed of sirolimus in its normal form, but carried by different agents, and those agents are in the single-digit micron range. You can see here the MagicTouch Sirolimus-coated balloon is really a phospholipid carrier that encapsulates the sirolimus, and they're submicron, 0.4 micron particles that are really spray-coated onto the balloon, and they give good adhesion to the drug, and I'll show you that in the PK data. You can see early penetration, and it gets into the deeper layers of the vessel wall. This is just a schematic of how that looks in terms of the way the balloons are made. You can see the phospholipid particles there and the encapsulating the sirolimus. Now, we've done a lot of studies with MagicTouch. We've done probably more than anybody else in the preclinical arena in terms of all the indications they've received for almost every indication you can think of. You can see that this was a comparative study. We compared POBA to MagicTouch to solution to paclitaxel agent-coated balloon in the porcine coronary artery, and we looked at the downstream emboli in this paper to see which drug-coated balloons were associated with embolic debris. And you can see in terms of myocyte necrosis scarring, that is areas of the heart that got scarred, that was exclusive to the agent paclitaxel-coated balloon, consistent with the embolic debris I just showed you. Both the POBA, MagicTouch, and solution had zero scarring. And in terms of single to multiple downstream emboli, now I should explain, emboli are different than scarring. You can have emboli and no downstream skeletal muscle or myocyte death, and that's the difference. You just see emboli. And you can see the emboli really line up in accordance with what I showed you in the scarring. There's the most emboli for agent and the least for POBA, and MagicTouch and solution are shown right there. Now, what about the pharmacokinetic levels of drug with these sirolimus-coated balloons? It's extremely important to understand that this is an important predictor of success because you need high enough drug levels. When they were compared in this porcine clinical study, coronary study we did, you can see here that the MagicTouch and solution had almost identical levels of drug at 28 days. Paclitaxel had a little bit higher, but you can see the spread was incredibly wide. And then drug in downstream myocardium, again, the paclitaxel-coated balloon had the largest amount of drug in downstream myocardium, with MagicTouch and solution having relatively little drug. Now, this was a study I conducted just recently, comparing the solution, MagicTouch, and Xience in porcine coronaries to look at the pharmacokinetic profile of these different sirolimus-coated DCBs compared to a DES, the gold standard DES Xience. And what I'm showing you here is the kinetic profile over time. And you can see in black is the Xience, basically a very steady state amount of drug in vessel wall, with solution having somewhat similar, although not as good levels. And MagicTouch has initially very high drug levels, which gradually decreased out to 90 days. And this is the downstream drug in myocardium shown here, showing again that Xience had no drug in the downstream myocardium, whereas solution and MagicTouch had a little bit more. Now, in terms of the histology of these in animal models, you can see that the MagicTouch and solution are almost identical in terms of the histological scoring that we produce in animal models. Injury scores, muscle cell loss, proteoglycan score are almost identical at 28 days in terms of the way these balloons perform. And the reassuring thing is no inflammation, nothing that you would be concerned about in terms of vascular reactivity or adverse effects. And you can see in terms of the PK data, the blood levels come down very quickly in hours after the balloon is deployed. And the treated segment iliofemoral has detectable drug out to 90 to 180 days, which is pretty reassuring and perhaps backs up the data they've had clinically. And the drug in downstream vessels, unlike what I showed you with paclitaxel, you can see it's barely detectable at 24 hours if you look at different skeletal muscles and you assay for drug levels. Reassuring. Now, what does it matter in terms of PK? What level are we looking for? It's actually not exactly clear what drug level we need. So I would like to just remind you that when we show these drug levels, the biological drug needed is not always clear. So in conclusion, MagicTouch is a submicron phospholipid carrier. The PK profiles indicate detectable levels of drug for 60 to 90 days. And the one-phase extraction shows nearly identical levels of drug to the solution. And myocyte necrosis and scarring, I think, is really superior with sirolimus-coated balloons in terms of what I showed you and the products tested as compared to the paclitaxel-coated products that I showed you earlier. Emboli were much less frequent, and these studies really do support the safety of this product over available paclitaxel DCBs. I think it shows you that MagicTouch will play an important role in coronary and peripheral applications going forward. I do understand that there's going to be a BTK trial released by MagicTouch at the upcoming ACC meeting. So with that, thank you for your attention.
Thanks, Luke. Look, why don't you grab a seat and join us for a second? Just to sort of distill the data a little bit, we have a couple of minutes, Brian, for discussion. But so in terms of drug levels, this is a question that keeps coming up over and over again. So the bioavailable drug, is it the same between the balloons, or is it different?
I would tell you that it's a complicated question. The single phase extraction of drug does show that there's probably better, I showed this yesterday in my talk, there's slightly better sustained release for solution over MagicTouch. There's no question. But is there enough drug to cause a biological effect over time? I think there is. And that's borne out by the data that we've seen so far versus things like plain old balloon angioplasty, the Sirona trial. I mean, there is clinical data supporting the efficacy of the balloon over PTA. I would tell you. Now, is there a sustained drug release like there is with a Xience stent? It's just not. That's just the reality. And so I think we're looking at a product that works and that is improving, but can't compete yet with stents. And we've seen that even with the solution trial, ISR trial that was released at TCT. If you look at the rate of recurrent ISR in the solution group versus the drug-eluting stent-treated patients, it's roughly double.
All right. Any questions for Alok about drugs? Go for it.
Yeah, hi, Sayal. My question to Alok is, we generally talk about the dose and the amount of drug, but what about where the drug ends up?
Yeah.
You know, in the media, adventitia, or endothelium? Is that important, and has that been studied?
I think it's very important, Eddie. Your comment is very on the money with that. And I would tell you that how the PK data is done does not tell you where the drug is. It only takes the entire segment and grinds it up and says, in this entire gram of tissue, we have this much drug. We do not know the location of the drug. You can do studies using fluorescent markers to show you where the drug is, and it's been done with MagicTouch, and it seems to evenly partition in the whole artery, consistent with Elazar Edelman's data for sirolimus.
Yeah.
I have one more question regarding the differences of the encapsulation and also in comparison to what we know from the drug-eluting stents. I mean, the sustained release on a drug-eluting stent is achieved usually by a polymer, right? So this is kind of mimicked by the solution approach, I would say. And in the MagicTouch, they're avoiding the polymer and they're using the phospholipid. Is there any downside, a potential downside to the use of a polymer?
Well, the potential downside is that the polymer itself can cause an inflammatory reaction. So like for Absorb, for instance, it's a polymeric scaffold. You saw that the inflammation curves for Absorb actually peak during peak degradation. Now, the difference, Dirk, is that, of course, with Absorb, you've got a large amount of polymer in the wall that's sitting there for over time. So you've got to degrade all of that. With Solution, remember, we've got polymeric transfer, but we've probably got not that much transfer. So we don't have nearly the dose of polymer getting into the wall that you would have with a bioabsorbable scaffold.
And have you seen any inflammatory reaction in any of the pathologic specimens? It didn't look like it from what you showed.
You don't, I mean, the 28 data is probably not the right time point to show you that, Sahil, because in what the FDA wants to see is they know that sirolimus is an anti-inflammatory. So during degradation, while sirolimus is there, you oftentimes mask the inflammatory response. You've got to look at the end of degradation, the end of drug levels, to see what's left in the artery. I didn't show you that data, but both these balloons have passed the test, they've gotten approvals. I will tell you the vessel is at complete quiescence at the end of drug release and looks very good.
And that's different than paclitaxel.
I would say that it can be different. It depends on the balloon. It depends on the kinetics. It depends on, what you see. I think with something like an alluvia, you aren't going to see. You're actually going to see an increase in inflammation over time. We showed that in preclinical models as the paclitaxel is released. It's a little more attenuated for drug-coated balloons. They don't have nearly the loaded dose of paclitaxel. So it really does, it's an important question that needs to be answered also in the clinic. How do the long-term data look with these different balloons?
All right, that's a perfect segue, I think, to now bring us back to Brian, who's going to tell us about the landscape of the other DCBs that we have in the United States as an example, and where sirolimus-coated balloon might fit in.
Thanks a lot, Sahil. So yeah, I'm going to be discussing the U.S. landscape, which is a little different than Europe. We have a bit more limited options, and we certainly have nothing below the knee just yet. These are my disclosures. So the first experience we have in the U.S. with drug elution for lower extremity disease is really with the Zilver PTX trial. It's not so much drug elution as much as it is a drug coating on the Zilver stent that's been shown to have good outcomes out to 5 years compared to PTA alone. True drug elution came with the Alluvia stent, which, as we've just been discussing, it's a polymer paclitaxel combination on a stent, and this also has been quite effective and quite useful in our lower extremity interventions. The problem with a combination of a drug and scaffold, however, is that the application of drug to the lesion really requires obligatory coverage of the entire lesion with a permanent implant, and many of us have seen the problems with that, and so we've really moved away from this idea of covering an entire lesion with scaffolds, and this full metal jacket approach is not something we typically want to do anymore. So luckily, at the same time that the various researchers and companies were investigating drug-eluting and drug-coated stents, the drug-coated balloon market has been explored. And one of the realizations is that you can actually bring in a balloon into an artery, inflate it for only a couple of minutes, and actually get effective drug transfer to the wall. As we've talked about, this really was first looked into with paclitaxel, or at least those are the devices that we have in the U.S. now. And there are a number of devices following a number of large randomized trials that have shown efficacy above the knee. And so these are some of the devices, or these are all of the devices we have for the lower extremity in the U.S. And the various IDE trials or randomized controlled trials that got approval for the devices, beginning with the Levant 2 study, which got approval for the Lutonix balloon, followed by the Impact SFA series of trials and manuscripts to support the Impact SFA DCB, followed by Illuminate and then Ranger. So that led to approval in the U.S. of a number of devices that are shown here, three of the four of which are shown here. And what I would say about these devices is I think they're variably effective and have variable efficacy, but they clearly all work. They all help achieve better primary patency than we see with PTA alone. And some of them have data out to 2 and even 3 years. And so this has been a real plus for us lower extremity interventionalists who are trying to keep vessels open longer, reduce the need for reintervention in our patients, and not only resolve symptoms of claudication, but ultimately save limbs in our CLTI patients. So it makes sense that we'd want to sort of use this above-knee device inventory in the below-knee space. And looking at what we have available in the U.S., again, these are the 4 balloons that we have available in the U.S. There's a number of other balloons that are available throughout the world, most of which we don't have in the U.S. One of the issues with all of these is they're all paclitaxel-based devices. I show this slide simply to point out that this discussion seems to continue to crop up of PTX mortality concern. I will tell you in the U.S. the concern is basically gone. I don't think our regulatory body has any interest in revisiting this. The work that Dr. Suskemsi, Dr. Parikh, and others have done have really shown that the concern about paclitaxel-related mortality is probably a signal that does not reflect any clinical concern. And so this is not really an issue that we have. On the other hand, the bigger issue, I think, is lack of efficacy in those devices that have been studied below the knee. Beginning with the Impact Deep trial, this is a trial first reported here at LINC over a decade ago, and it basically showed a failure to meet the primary efficacy endpoint of the paclitaxel-coated drug-coated balloon. as well as an amputation trend in the patients treated with the DCB. And so this was concerning because it was the first large-scale trial trying to get approval of a device for the below knee space using a paclitaxel-based balloon that failed to meet its primary endpoint and then was ultimately shelved. Shortly thereafter, the Biolux P2 study was also reported, and this again failed to meet its primary endpoint without any safety concerns. But again, another failure in the paclitaxel DCB below knee space. And that brings us to the Lutonix below the knee trial. This showed some efficacy at 6 months by Kaplan-Meier analysis, but the primary predefined primary efficacy endpoint showed no difference between these two arms. And this device ultimately went to panel at the FDA and was not approved. And so again, we're left at this point in the U.S. without having any devices for the below knee space in terms of drug-coated balloon devices. So this kind of begs the question whether or not there's any safety concerns. The question is paclitaxel the right drug for below knee disease? It is a proven antiproliferative agent. It certainly has been shown to be effective in our SFA and popliteal vascular bed, but it simply may not be the right thing for the below knee space. It has a limited therapeutic window, probably some variable efficacy within that window, and as I just showed, it's really failed multiple trials. And so it makes sense that we should question whether or not there's another device or drug combination that we should explore. And that's where Limus as a sirolimus and everolimus may show some promise that our paclitaxel devices have not. This has a proven antiproliferative effect discovered back in 1970. It's been used and has become the dominant, Limus-based devices are the dominant devices used in the coronary circulation. It has a couple of properties that probably make it ideal for use in the below knee space. It does have a wider therapeutic window than paclitaxel, so the dose and clinical response interaction can probably be manipulated a little bit more than our paclitaxel devices. It does have, as we've discussed, some anti-inflammatory properties, so this could be a helpful component of these as well. And so I think this is a reasonable thing to look into, but there are some differences that we need to be aware of. Certainly with sirolimus, I mentioned some of the positives: wide therapeutic range, much better safety margin. It has some anti-inflammatory effects as well as maybe some better effect in terms of inhibition of smooth muscle migration and those sort of factors. On the other hand, tissue absorption is slow, tissue retention is short. This is something that is a little bit harder to get into the wall and into the vessel than our paclitaxel devices. And this could be looked at as a positive or a negative. One thing I'll say in terms of the paclitaxel, this has been sort of easier to get into walls and get delivered. There's been a number of different excipients that different manufacturers have used to help transfer the drug from the balloon into the wall. But I think this ease of transfer of the drug to the wall has left us with some sort of crude excipients, I'd say, in terms of getting that transfer to be achieved. And this probably is a reason why many of these devices that we have in terms of paclitaxel balloons do have some concerns with the amount of coating that can flake off, as Alok has talked about, and some of the concern with distal embolization after use of some of these devices. And so the sirolimus balloons, which I'll discuss in just a moment, do require a little bit more engineering to be able to liberate the drug from the balloon and get it effectively into the wall. But this also means we have some more advanced or second generation delivery systems. These are the clinical trials that are either under development or ongoing in the U.S. Concept Medical with the Magical BTK study, the Solution trial by MedAlliance, and Surmodics is also under development as well. In terms of the Magical BTK study, this is a one-to-one randomization of POBA versus PTA. POBA versus the MagicTouch balloon. It's ongoing in the U.S., Singapore, and Japan. This is, as I mentioned before, the coating requires a little bit more advanced coating than we have in some of our paclitaxel balloons. Because sirolimus is more hydrophobic, we have to coat the drug in a phospholipid layer, and this is just demonstrated here. You have a hydrophilic head and a lipophilic tail. This coats the drug in a way that allows for freeing the drug from the balloon into the vessel wall and uptake into the vessel wall. The coating is more uniform and less likely to embolize in large pieces as compared to some of our paclitaxel products. The device also has a unique patented coating mechanism in which the device is coated circumferentially at a low pressure inflation, followed by wrapping the balloon, protecting some of the drug within the folds of the balloon, ultimately leading then to inflation, giving a good drug delivery at time of inflation. There are two trials that are either ongoing or planned right now. We're in the middle of the Magical BTK study using this device in the baloney space, as I mentioned before. And we have a plan to begin the Magical SFA IDE trial in the U.S. as well. And that's under the site selection process at this point in time. This is the various details of the study and leadership teams of both the BTK study as well as the SFA study as well. And these studies will add to really the wealth of data that Concept has already begun accumulating. These trials shown here, some of which we'll hear more about in the next couple of talks. So in conclusion, drug-coated balloons have proven useful for PAD in the femoral popliteal segments and have reduced reliance on permanent implants. Multiple paclitaxel DCBs have been studied in the baloney space in randomized trials without demonstrable benefit. And so this new class of limus-based DCBs are now undergoing evaluation in large randomized trials for the baloney space. And hopefully these devices will have some additional properties that will allow them to be successful in these trials and ultimately prove efficacious for tibial disease. Thank you.
Ryan, thanks. That was a tour de force reviewing the space. Any questions for Brian from our panel? So in terms of understanding how to apply these technologies, we've got our panel fully fleshed out right now. We have online Dr. Samir Dani from India, who's some of the world's largest experience using MagicTouch in both coronary and peripheral arteries. And then up here, we also have Dr. Tatsuya Nakama and Masahiko Fujihara from Japan, who've joined us and fleshes out our global experience. Why don't we have a talk next by Dr. Schmidt, who's coming to us from the cath lab out of his lead for a few minutes, and we'll talk a little bit about how we're going to incorporate this into our treatment algorithm. Andre, good to see you.
Yeah, good to see you. Thank you. Yeah, thank you, Saeed. Yeah, thanks for inviting me to participate here in the symposium. Well, this is not going to show you here cases about using sirolimus-coated balloons in fempop lesions. They look similar to when we use paclitaxel-coated balloons. But this is maybe a summary of thoughts which an interventionist has sitting in the cath lab and using paclitaxel-coated balloons every day. Is it now maybe better now to change from PTX to maybe sirolimus or in the future? So these are my potential conflicts of interest. So in fact, after the Katsanos paper, we were, of course, very much relieved when we saw many other papers following that, showing that there's obviously no sign of a higher mortality using paclitaxel-coated balloons, as this one here just recently published, more than 160,000 patients where drug-coated paclitaxel-coated balloons were used. And you can see here that there was no signal of higher mortality. So in fact, seeing this, we thought, well, is the strong argument maybe using now sirolimus-coated balloons is not there anymore, but now then the paper here from Nordansky came, Sweet Batu, again showing this signal here of higher mortality using paclitaxel, and we are concerned again and do not really feel very well again, similar to the time when the first paper was released. So this systemic toxicity, which we cannot really see, the local toxicity, in fact, we can see in our cases here. We see these aneurysm formations around paclitaxel-coated stents. The Eluve here, for example, 8% aneurysm formation here in the publication from Münster, up to nearly 20% in this capsicum study from Japan here, when using paclitaxel-coated devices together with IVUS, so a more aggressive predilatation. This is certainly of concern. And also in our cases, we do see this sometimes, and certainly this we haven't seen before the area of paclitaxel. And this, of course, also has to then be followed by some other interventions, for example, in taking covered stents in or so. We also have seen this not only after drug-eluting stents, but also after drug paclitaxel-coated balloons and fempop lesions, has been occasionally published as this here, aneurysm formation here in the SFA, also aneurysm formation here below the knee. We also see these cases, especially after repeat treatment with paclitaxel-coated balloons, as in this case, where we had to open up here the anterior and the posterior tibial several times, aneurysm formation up to the, situation that it actually even caused an AV fistula here in that area. We see this also here after SFA treatments, maybe especially after subintimal passage. There are also reports and thoughts that it's even higher, this formation of at least aneurysmatic or dilatation formation after atherectomy using paclitaxel-coated balloons. Well, we always think this is not of a sequela, doesn't matter, but actually at the end, we don't know whether this at some time may cause some problem in terms of maybe acute thrombosis or so. So the acute toxicity is seen, and it is again, maybe more of a concern if we combine paclitaxel-coated balloons, maybe with a bare metal stent, as in this case here, treated with a paclitaxel-coated balloon, followed by the superior stent, the artery dilated, the stent is oversized, now swimming around, and occasionally may then be a reason for acute thrombosis. This is another problem we occasionally see using paclitaxel-coated balloons in our treatments here. Slow flow after the treatment, long SFA, total occlusion in the CLTI patients, very complex, was difficult to go through. But you can see here after treatment also with IVL, some bleeding after retrograde approach, no problem, was a good flow. We followed then, as we usually do with drug-coated balloons, 3 times 150 millimeter in length. And that's what we saw thereafter, a significant slow flow here without any gross embolization of atherosclerotic emboli, which resolved only after half an hour. So this is certainly sometimes of concern for us. It's well known that, and you have heard it, paclitaxel balloons embolize downstream. There's a report that this slow flow phenomenon is encountered around 8% after using paclitaxel-coated balloons in these kind of lesions, especially in longer lesions, poorer tibial outflow, and this was also slow flow associated with poorer short-term outcome. Maybe, of course, also because of the complexity of the lesion. But as far as we heard, that was never, in fact, the case with sirolimus-coated balloons to see this slow flow phenomenon. Fibrinoid necrosis after downstream paclitaxel embolization in animal models also has been seen. Maybe the link to sometimes reported higher amputation rates after using paclitaxel-coated balloons may also not be the risk using sirolimus-coated balloons. So what might be the advantages now then with sirolimus-coated balloons? It's less toxicity, potentially less local issues. Potentially then we would prefer it, especially after treatment using atherectomy, for example, after subintimal recanalization, for repeat DCB treatment, maybe also in combination with bare metal stents, and also in our CLTI patients because of this lack of slow flow, and maybe because of then a less negative impact on wound healing, having less embolization. Well, you know, this loss of drug occurs when you open the balloon, also when you take the balloon from the sheath down to the lesion. But whether the drug is also lost maybe by simply taking it out of the package and unsheath it, taking the protection sheath off, is studied here in this paper here coming from the group of Ulf Teichgräber from Jena. So they investigated the drug combination of the working environment with paclitaxel-coated balloons. They studied several balloons here and did photos with high-speed camera of the balloon surface during withdrawal only of the protective DCB cover in an environment where they could do this wipe test, collect the potential paclitaxel dropping off the balloon. They also did some air samples and sent it to an institute to investigate the concentration of paclitaxel. And they found that after removing the protective cover of the paclitaxel-covered balloon, surface was significantly reduced already in some of the balloons, was just lost in the protection cover, obviously. But in 6 of these 7 balloons, powdered paclitaxel also dropped down. It may drop to the floor on your shoes, I don't know what. No air contamination, however. And well, the reduction in the therapeutic dose and contamination of the workplace, well, is there possibly a hazard for medical personnel? We don't know. So in summary, I think if paclitaxel and sirolimus-coated balloons would lead really to similar results in our patients with infrainguinal treatment, sirolimus-coated balloon could really be the preferred choice for the patient, maybe also for our staff in cath lab. Thank you.
Thanks, Andre. Let's have a little bit of a discussion before we get back into more data. So we have a number of users of the device here who have lots of clinical experience. I know Dr. Dani is on from India in the evening time there. Samir, how are you? Can you hear us?
I can hear you.
Okay.
Thank you for having me there.
And
it's been a very interesting discussion.
You've used this device in both coronary and peripheral arteries for probably longer than just about anybody. Tell us a little bit about your experience with seeing no reflow, and have you really experienced any of that with any of these devices?
Yeah, I don't remember having seen no reflow with sirolimus-coated balloons, MagicTouch especially, because that is where my maximal experience lies. Not a single case I remember where there was no reflow following the MagicTouch. I think that is particularly of importance in BTK arteries, where or critical limb ischemia patients, where prolonged inflations, high pressure inflations because of the calcification you see in BTK and the fragility of the arteries and smaller arteries, this may be particularly important advantage over paclitaxel-coated balloons.
Yeah, Eddie, you've had a similar experience, right? You want to amplify that a little bit?
Yeah, absolutely agree with Sunny. With MagicTouch Sirolimus-coated balloon, you never see any evidence of slow flow phenomenon, even if it's deployed or applied really distally into small arteries, including the plantar arch. So we never see that. Whereas conversely, with paclitaxel, the thing which is always on my mind before I apply it is whether I'm going to get slow flow phenomenon and if I can get out of it. Most of the time you get out of it, but I've had 2 occasions where there was distal vessel thrombosis, which I couldn't salvage. So,
yeah, I just want to add that I think this issue of distal embolization is one that is probably under-recognized, and we have a session tomorrow on distal embolization and the scope of that problem. And it's very clear from the medical literature that patients who undergo secondary bypass after a failed percutaneous intervention have worse outcomes than those that went straight to a primary bypass. And there could be a lot of reasons for that, obviously. I mean, some of it is a selection bias. You know, these may be bad actors, but there's probably a high likelihood that many of these patients are suffering microscopic distal embolization that we just don't recognize. And slow flow phenomenon or no reflow is one problem, but that is a very severe problem. We probably have a much less severe microembolic risk every time we do anything. And if you look at transcranial Doppler probes placed on a popliteal artery, there's been at least one study in the medical literature looking at that. It shows that at every stage of an intervention, even wire crossing, you're going to get embolization. And so you can imagine that when we can catch particulate matter in a filter from these balloons, this problem is one that is likely under-recognized. So anything we can do, I think, to minimize that risk is probably worth doing.
Yeah. Tatsuya, you saw the same in your capsicum studies, right?
Yes, as Ryan mentioned, so it's we always underestimate this distal embolization phenomenon. It's slow flow is only a phenomenon. It's not always a result from the embolization. It's completely depending on the condition of the lung officer. We often see this kind of the phenomenon, especially for the very poor lung vessels in serious patients, CLTI patients. Also, we sometimes see is a very poor lung of the coronary arteries. Therefore, we more focused on the embolic particles, and therefore, I think we have to care much about of it.
Andre, in your experience, do you pre-medicate patients with nitroglycerin or any other vasodilators now more routinely, or have you stopped having to worry about that now that you're using MagicTouch below the knee?
Well, I mean, yeah, that's difficult to say. I mean, it's absolutely standard for us to use nitro in below-the-knee treatment, so cannot tell you whether it's we see some other results using no nitro, so it's actually standard. But not sure, yeah, maybe it diminishes the risk of no-flow, reflow using paclitaxel balloons. I don't know exactly.
Yeah.
Sorry.
I mean, I'm not so sure actually about all these vasodilators, because at the end of the day, if you embolize solid particles, crystalline material, I mean, that gets stuck somewhere and it causes slow flow. And, if you give vasodilators, I mean, what are we doing? I mean, just probably pushing it one level further down. Right. But we are not eliminating, we are not resolving these particles. So I think this is something what we surely underestimate, and that's why I also put the session for tomorrow on the program, because I truly believe it needs more attention because we need, particularly in the CLTI patients, I mean, we speak so much about the distribution to the actual wounds. And if these, particles get in between, this will not give us a good success at the end of the day. So we need better solutions, and we need better strategies to avoid it.
Right.
Can I add one thing on that?
Yeah.
I think that where these lodge are so small, the arterioles, that I don't think a vasodilator is going to do much, because they really are, you can see them there, and the surrounding necrosis that occurs, fibrinoid necrosis so-called, we call it, because it's fibrin that's produced by the paclitaxel, and it basically causes inflammation around the arterioles. You sort of see a ring of inflammation around those arterioles. So I don't think the vasodilator, I agree with Dirk, is going to do much. And I also point out that all of our studies that we've done in these controlled settings are very different than what you're doing in the clinic, because the flow rates in a healthy animal are just not the same as what you do when you do these interventions in people with poor outflow. I mean, I think the impact is exaggerated in the clinic.
Yeah, to both of your point, you can use all the vasodilators you want. You're not going to turn a capillary into a 2-millimeter vessel. And I think it's worth recognizing that we're all seeing increasing role for such procedures at deep venous arterialization, specifically because we have a lot of patients with no outflow in the foot, and some of that is their own primary disease, and some of that may be what we're contributing to.
Yeah, so if there's a safety issue at all, it's embolization as much as anything, and prevention would be the best treatment. And so that's maybe one of the biggest roles for sirolimus-coated balloons. So let's switch gears and talk about efficacy, maybe. Dirk, do you want to give us an update on how sirolimus-coated balloons are being applied in PAD in our clinical trials, Sirona and Limus?
Yes, absolutely. So I assume these are my slides. Yeah. So I want to give you a brief update on 2 trials, which both have been investigator-initiated trials run here in Germany. One dedicated to the to the femoral, and one to the below-the-knee circulation. Well, the Sirona trial has been meanwhile presented a few times, and still, I think it's very important, because it's really the first head-to-head comparison of a sirolimus-eluting and paclitaxel-eluting drug-coated balloons in the femoral popliteal segment. As I said, was investigator-initiated, run with a grant support from Concept Medical. 25 sites were recruiting in total 482 patients, and those were randomized 1-to-1, as you can see here. And the primary endpoint reading was after 1 year, but of course patients are monitored much longer, and I'm able to show you today the 2-year results, and the 3-year results will become available relatively soon. Here are some patient demographics. So predominantly claudicants with Rutherford category 3 and 2 in this cohort. If you look at the lesion characteristics, a lesion enrollment was allowed up to a length of 20 centimeters. That resulted in a mean lesion length of around 8.4 centimeters, which is relatively long for such a randomized clinical trial. What is also of note is that the rate of chronic total occlusion is 34%. And 32%, and also the degree of calcified lesions, PECs 3 and 4, was really relatively high. So clearly a somewhat challenging cohort for a randomized clinical trial. That's also reflected here by the procedural characteristics. Not all of the lesions could be treated perfectly well just with a balloon alone. There were some dissections, some severe dissections in both groups, and the bailout stent rate was in the range of 24% and 21% in the group. So I think this is still in line with other studies. So actually probably rather low for this relatively complex cohort, but of course it was necessary in about almost 1/4 of the patients to perform some sort of spot stenting. So what about the primary endpoints? Primary efficacy was measured by primary vessel patency based on duplex criteria with a PSVR of 2.4 and the absence of target lesion revascularization. And the primary clinical endpoint was a composite of a clinically driven target vessel revascularization, major target limb amputation, and device or procedure-related death. You can see here the primary patency endpoint at 1 year met the non-inferiority hypothesis with a significant p-value, and you can see the primary patency based here on this nominator-denominator method was in a range of 74 to 75% in both groups. The primary clinical endpoint was also met, showing non-inferiority with regards to these mainly safety aspects, with around 90 to 92% freedom from these composite events. And you can see that predominantly, or I should say almost exclusively, these were target vessel revascularizations. And if you see if they were adjudicated for being for the target lesion, and then these were of course fewer events. So I promised you to show you the 2-year results. This is the primary patency out to 2 years. You can see that both balloons performed equally well with a primary patency of around 67 versus 65% at 2 years, and non-significantly different. And if you look at the TLR curves, you can see that the sirolimus-coated balloon was doing a little bit better with 91% freedom from TLR versus 88%. So again, no statistical difference, but I think respectable results if you consider the complexity of the cohort. Major amputation was not an issue in both groups, as you can see here, and also freedom from death rate was not different and very high. I think very typical for a claudicant cohort. So how do the results look like from a patient perspective? What were the measures here? We employed a 6-minute walking test in the protocol, and also the vascular quality questionnaire. You can see here to start with the vascular score. the questionnaire, there was a significant improvement versus baseline in both groups, and no difference between the groups. As measured by improvement of Rutherford scales. Also, the clinical improvement was very pronounced. Almost 90% of patients had at least one Rutherford category improvement, and we didn't see any differences here. And also the 6-minute walk test showed improvement in both pain-free walking distance, absolute walking distance, and no statistically significant differences between the groups. So I think we can say that, well, first of all, the sirolimus-coated balloon demonstrated the non-inferiority at 1 year, which was the primary objective of the study. And at 2 years, we can still say that we don't see any differences in regards of, well, anything, I should say, neither primary patency, clinically driven target lesion revascularization, or any clinical endpoints, including major amputation, death, quality of life, or walking distance. So that's positive, and we are looking forward to see the 3-year results soon. Well, clearly, that was about intermittent claudication and femoral disease. If we speak about chronic limb-threatening ischemia, that's a completely different story. We are looking clearly at older patients, more diseased patients, and predominantly also an involvement below the knee. That's why the so-called LIMUS study, again, a physician-initiated study conducted here in Germany, was set up in a somewhat different way, because we had to realize that current standard of care for below-the-knee treatment is probably POBA, plus minus some provisional stenting with bare stents. Well, so far, none of the balloons really could show a clear superiority. At least I would say the evidence is contradictory for paclitaxel-coated balloons, and the long-term safety is also not absolutely clear. So from that perspective, sirolimus is an interesting drug, and in its formulation on the MagicTouch, and probably an interesting opportunity, which we decided to study in the LIMUS trial. That's another piece of uncertainty raised by Dr. Katsanos. He analyzed the below-the-knee randomized clinical trials, and as you know, he showed that there was an increased risk of amputation in these trials. That's at least something which has to make us alert. So the LIMUS trial randomized again in a 1-to-1 fashion, and total 230 patients, 115 participants per group at 28 sites in Germany and Austria, as you can see here. The primary endpoint will be measured by a composite of limb salvage and primary patency at 6 months. And the primary patency is defined as an absence of target lesion occlusion with restoration of inline flow to the ankle without the need for clinically driven target lesion revascularization. All that will be assessed by core labs, of course, and then we will also assess secondary outcomes like Mayo and so forth. The main inclusion criteria are Rutherford 4 to 6 patients with infrapopliteal target lesions. We focus here on total occlusions, and single or multiple lesions are allowed. And yeah, you can see here one of the typical patients which we treated in this trial, 79-year-old female patients with rest pain and on the right forefoot, reduced ABI, and a list of comorbidities, presenting here with this typical long occlusion of the tibial vessels, the anterior tibial and the posterior tibial. The anterior tibial was treated as a target vessel. You can see that retrograde approach was needed, so that's allowed in this clinical trial. Eventually, after the card technique, the crossing was successful, And then the vessel was pre-dilated and finally dilated with MagicTouch balloons of 2.5 millimeter distal and 3.0 proximal, as you can see here, with a good result and a good inflow into the foot. So the LIMUS trial really enrolls full-length lesions. You know that in many of the FDA approval trials, we are sometimes limited with lesion lengths. This was not the case here since we are using a device with a CE marking. So we are really able to take on the lesions which we see on a daily basis. And that's probably also one explanation while this trial enrolled reasonably fast, still challenging as every BTK trial, but you can see here that the target of 230 patients was reached after roughly almost, I would say, 2 years, no, 3 years actually. So yeah, I mean, it was still challenging, but I think we have it now. The follow-ups are coming in and the analyses are ongoing. So I think later during 2026, we will see the first primary endpoint results of this trial. Thank you.
Great. Thank you, Derek, very much. And Eddie, why don't you come up and tell us about the HOPE registry, sort of dovetail with that, and then we have some interesting additional info to share with the group about other trials.
Thank you very much, Sahil. So I will now present the updates on the 4-year results of the BTK subset of the HOPE registry. The data was first released in yesterday's symposium as well. So these are my disclosures. The HOPE BTK registry is a prospective registry of all lower limb angioplasty procedures conducted in a single center in Singapore. The aim is to collect data and analyze the clinical outcomes of MagicTouch Sirolimus Coated Balloon. And we also have a comparator group in patients who receive POBA only. So we included patients with CLTI, Rutherford scores 4 to 6, de novo lesions with BTK lesions treated with MagicTouch Sirolimus Coated Balloon versus POBA. We excluded patients who received paclitaxel devices, thrombolysis, or atherectomy device, and we have a population of 133 patients with MagicTouch Sirolimus Coated Balloons versus 185 patients with POBA-only strategy. So patients were enrolled between August 2018 and October 2021, and we now have data on 4-year clinical outcomes, including freedom from target lesion revascularization, freedom from amputation, and also the overall survival. So first of all, let's have a look at the demographics of these two cohorts, Sirolimus Coated Balloon versus POBA. In terms of age, these were on average about 70 years of age. Majority were male. In terms of the medical conditions, a typical CLTI cohort, 90% diabetics, with a large proportion of patients with significant coronary artery disease. And up to about 20%, roughly about 20% were already on dialysis, having end-stage renal failure. And these two groups were quite comparable with no statistical significant difference between the two groups. In terms of the ASA scores, again, reflects the highly frail, high-risk patient group. Majority were high ASA scores as judged by our anesthetists. In terms of the Wi-Fi score of the wound, this is also considered quite a high mean score of the wound, with an average of about 3.9 to 4.1, similar across the two groups. If we look at the lesion characteristics, majority had moderate calcification, but I would also like to point out that in terms of moderately severe and also severe calcification, there was also quite a large number, maybe numerically more in the POBA group, but the p-value was not statistically significant. About a quarter of patients required retrograde access. Lesion lengths were long. We calculated the total lesion lengths of all the tibial arteries treated, and this amounted to about 235 millimeters in the Sirolimus Coated Balloon group versus 250 millimeters in the POBA group. So now I'm going to move on to the results at 4 years. First of all, the freedom from target lesion revascularization. At 4 years, sirolimus-coated balloon usage was associated with a 39% reduction of BTK reinterventions. In absolute numbers, the 4-year freedom from target lesion revascularization was 75% in the MagicTouch Sirolimus-coated balloon group versus 62% in the POBA group, so a delta of about 13%. So if you look at the graph, you can see that from 0 months to 5 months, the graph were quite similar, and it starts to diverge at about the 5 months, 6 months point onwards. And it continued to diverge, and we saw this effect firstly at 1 year, 2 years, and also 3 years. The delta then was about 10%, and we wondered whether at 4 years it was going to converge, but it did not. In fact, it did the opposite, slightly diverge a bit, and so the delta was slightly higher at 13%. So we also adjusted for comorbidities, and we found that other risk factors which predicted for target lesion revascularization was severe calcification and also multi-level disease in both above the knee and also below the knee. We next looked at survival, and this is somewhat surprising to us. At 4 years, sirolimus-coated balloon was associated with a 32% increase in survival. We first saw this effect at the 2-year mark and also the 3-year mark. And we wanted, when you look at the graph at 6 months, the survival was quite similar, and it starts to diverge at about the 6 months mark. So quite closely correlates with the amount of reinterventions that we were doing. So we postulated that the advantage in survival was most likely, and this is an extrapolation, we do not have any statistical data for this, in that we think the advantage in survival is due to the lower number of reinterventions in the sirolimus-coated balloon group. And at 4 years, you can see that majority of these CLTI patients had already died in the POBA group, only 43% surviving versus 55% in the MagicTouch Sirolimus-coated balloon group. And again, this was adjusted for comorbidities, and we saw that other risk factors in our patient cohort which predicted for mortality was age. Obviously, I mean, this was highly statistically significant, p-value of less than 0.0001. Female sex was also a predictor for mortality, and this is consistent with other studies with CLTI patients. And also, the more severe the wound, the higher the risk for mortality. And again, this is consistent with other literature. Lastly, we looked at freedom from amputation. And at 4 years, the freedom from amputation in the sirolimus-coated balloon group was 92% versus 81%. However, this was not statistically significantly different, just numerical advantage, not statistically significantly different between the two groups. There were other risk factors which were much more predictive for amputation, and that's end-stage renal disease patients and also patients with severe calcification. And again, this is consistent with literature and obviously consistent with our own anecdotal experiences as well. Patients with kidney disease, with highly calcified vessels, are the hardest patients for limb salvage. So in conclusion, this is a real-world experience of our experience on MagicTouch Sirolimus-coated balloons for BTK lesions in CLTI patients. And personally, I believe this is where sirolimus-coated balloons is really advantageous, BTK lesions in CLTI patients. And at 4 years, in our experience, the sirolimus-coated balloon was associated with better freedom from TLR, 39% reduction in BTK reinterventions, better survival, 32% increase in survival. However, for amputation, although there was numerical advantage, it was not statistically significant. We found that patients with kidney disease and calcifications were highly predictive for amputation. Thank you very much.
All right, it's really compelling sets of data. Ramon, I wanted to get your thoughts initially to Eddie's presentation, where he's able to show survival advantage as well as patency advantage. What, having been involved with you guys on a bunch of these trials, this seems like an extraordinary result.
Yeah, I mean, congratulations, Eddie, on putting together another data set, one of many. I was pretty taken aback by the high mortality rate in the PTA arm. And I think that probably reflects the complexity of patients and comorbidities that you treat in Singapore. I think you guys treat the sickest patients around, end-stage renal disease and often desert feet and that sort of thing. It's hard to sort of interpret that difference without a randomization process. There's bound to be some selection bias in there. It is a little bit hard also to interpret why patients would die more often. Certainly there's a patency difference that we'd expect if the balloon's efficacious, but could that cause mortality? It's hard to really understand that. But I am equally as excited by the prospect of this device in BTK arteries. I think this is the unmet need. We've got pretty good femoropopliteal solutions at the moment. Sure, they can be improved. I think we've heard all about that in this session. But in BTK, we don't have great solutions. We've got scaffolds, but to treat these 30-centimeter CTOs, they're largely impractical. So it'll be fascinating to watch this play out and see the role that sirolimus-coated balloons might have, perhaps even in combination with scaffolds, to treat this long pattern real-world disease.
Yeah, Masahiko, in Japan, you have a high burden of renal disease and calcification.
Yeah.
So this is a patient population that I assume is similar to what you're treating for CLTI.
Yeah, thank you very much. Very good comment. So yes, in Japanese real world patient population have the very, very high prevalence of the diabetes and end-stage renal dysfunction and very severe calcification. So yes, added data is a very, very emphasize that for real our patient background is how to deal and how to manage these regions. Our next challenge. So unfortunately, we don't have any DCB for the BTK regions. So one question to Eddie. You have so many experience of the BTK regions after the MagicTouch Sirolimus DCB. So are there any difference to the restenosis pattern? So the thrombotic reocclusion or the spots restenosis or the long diffuse restenosis. So how about the restenosis pattern after the MagicTouch DCB?
I don't see any difference in the restenosis patterns. When they come back, in our experience, they are pretty much the same as POBAR. So you do still see, to be honest, just the diffuse stenosis and reocclusions that you see with POBAR. Yeah.
And
so I do not know why some patients are less responsive to MagicTouch Sirolimus-coated balloons and why they restenose, but their patterns are similar.
And are there any concern of the aneurysmal degeneration, similar to the paclitaxel device?
No. I've never seen any aneurysmal degeneration with sirolimus-coated balloon, and I've very rarely, if any, I don't remember seeing any slow flow phenomenon as well.
Thank you.
Can I ask Eddie, when you look back at the deaths in this registry, do they seem to be related to reinterventions? Ramon and I were kind of commenting on the very high mortality rate in the group overall. It's not inconsistent with CLTI patients, but it's pretty high, and it does speak to the fact that even if you don't see a direct complication related to reinterventions, those reinterventions take a toll on these very frail patients. Even if it's putting them in a hospital for a day or two and taking them out of their home environment, that can have ramifications in terms of pulmonary infections and mobility and all of that. So did you see any direct relationship to the mortalities and the deaths and the reintervention?
Yeah, I think thanks very much for raising that important point. I think we need to see, in order to understand, you need to understand the way we treat CLTI patients in Singapore, in that we admit them, all patients are done under general anesthesia, and they probably stay in hospital for another 2 to 3 weeks for us to manage the wounds inpatient, and they develop a lot of complications. So TLR and reintervention in our setting is not the same as just a procedure and you send them home the next day or even the same day. So it is quite an extensive procedure that they undergo, and so I think that's why intuitively I think that adds on to the mortality if you were to have more reinterventions. In terms of the cause of death, the majority by far are cardiovascular deaths, so heart failure, MI, and things like that. Yeah.
All right, well, as we wrap up here, just to summarize the clinical data that we've heard about, we now have real randomized head-to-head data against paclitaxel. There's no other sirolimus-coated balloon with that data in the peripheral artery circulation, especially in the FEMPOP level. We have forthcoming randomized against balloon angioplasty data from this data set, and I just got a notification that the SIRPAD trial has been accepted as a late-breaking clinical trial at the ACC, which is in March. And so there will be randomized data in that group for both above and below knee patients with both claudication and critical limb ischemia. And so we'll have, again, real randomized data against a real comparator very soon. So we'll see if our signals that Eddie has highlighted may come true in prospectively randomized trials. So there's an avalanche of data forthcoming in the next couple of months. With that, any questions from the audience would be welcome. And seeing none, I want to thank our sponsor, Concept Medical, for a terrific program, and I want to thank all of our panelists for participating. Dr. Donnie, thanks for sticking with us this afternoon, and we'll look forward to seeing all of you at the rest of the meeting. Thanks very much.
Thank you.